for joining us today for the July 30th edition of PopBio on the Dark Side. Just to repeat the theme of this seminar series, we're essentially just inviting speakers in the field of broadly population biology to talk about their research. Happens once a month, usually on the last Thursday of each month. In the chat, I will post a sign-up sheet. I assume everybody probably already has access to that here. But also, if you're willing to, please share the link with others in your department or lives who might be interested. And so today, I'm pleased to welcome Chang-Yu Chang, who is sort of a recently repatriated member of the dark side of ecology in this planet. He recently relocated to Academia Sinica, which is in or just outside of Taipei, Taiwan, to work at Academia Sinica, which is a really cool, like, interdisciplinary research institute that I had the pleasure of visiting a few years ago and was just super impressed by all the research going on there and the breadth of research. And so before that, he was a postdoc at the University of Pennsylvania, working on plant microbe symbioses, and did his PhD at Yale University with Alvaro Sanchez, working on microbial community ecology and sort of the interactions in these complex multi-species communities. And so you can read his biography online, but it seems like he's really interested in sort of the ecology and evolution of plant microbe symbioses. And when we met a few months ago, he took me around and showed me some cool plants that he's going to adopt into his study system that are these, unique little nitrogen fixing forbs. So maybe we'll hear about that as well. And just to remind you, we are recording the seminar, but we will not record any questions that happen afterwards. So please feel free to stick around and ask questions, introduce yourself to the group. Okay, with that, thank you, Chang-Yu. Please take it away.
Yeah, thank you. Can you all hear me good, right? Great. Thank you, David. For today's talk, I would talk about part of my PhD, part of my postdoc, and also spend a little bit of time about the future outlook and just introduce amazing plants that I found native in Taiwan that has the potential to answer questions I'm interested in. And one thing that I kind of pick up from David's introduction is, so Academia Sinica, it's technically in Taipei. Like if you look up, the district, like it's in Taipei, but it's in a remote place. And recently, like attended a kind of like a special exhibition in Sinica for like agriculture and history of Sinica. And I always wonder why is it in such a remote place? It's in Taipei, but not the Taipei you think of. Like it's not very dense in population density. And apparently the reason that's the first president of Academia Sinica decided to choose this place because it's such a remote and quiet place. So it's a perfect location to do research. I enjoy here. So, all right. So a little bit about myself. So I will see myself evolution ecologist, and I started my interest in like kind of complex system and as an undergrad, and then got kind of interested in animal behavior always has been interested in insect behavior as a kid. So I did a study on burrowing beetles behavior. Happy to talk more about that later on if I have any questions. And then one thing I realized when I was working on these social insects is that microorganisms, I think I will argue that it's the most social organism in the world because you never see one microbial cell living alone. It's always with other cells, right? So I started to kind of work on microbes in PhD, and then that was kind of started to think about community ecology, and also I guess most of the people I know work on microbiology experiments in PhD. They start to brew their beer or like kombucha or make their sourdough. So that's the picture of the beer I brew during COVID. And then I got interested in host-related microbes and started to do some legume rhizobia studies, which I'll talk about later, and then kind of delve into the world of genetics or population genetics, evolution genetics. and kind of combining these interests in the past, kind of making the path forward to like what I'm thinking about on the upper right, some ideas of symbiosis, but thinking about more kind of multiple microbes, and also kind of taking advantage of the great natural history and then the collection of specimens to kind of study symbiosis in a native system in Taiwan here. All right. So as I'm sure all of you in the audience will agree that biology is very complex, and then uh, through the interactions of cells, tissue can function more than sum of cells. And it's throughout the biological organizations from molecules to ecosystems. And this is what we call emergent properties, like the title I have in this for this talk. And in principle, it's 1 + 1. It's greater than 2. And of course, microbes or microbial communities or any communities are not exceptions. And in biochemical, geochemical cycles, or the flavor of stinking tofu or kimchi or sourdough, they are all flavors come that been metabolized and digested by millions or billions of cells, and then making these complex flavors that we're familiar with. And I think one of the major aspiration in modern biology is to understand how we could kind of understand and, in principle, engineer or design the functions of these microbial products toward our desire. So, one thing I will argue is that this emergent property, so-called emergent property in microbial communities, arises from the ecology evolutionary context. And what I mean is that all these species, microbes, they are not independent. They are bounded by their evolutionary past and also what kind of ecological context they are in. And for examples, they will have interactions with other microbes. They depend on each other and the environments for their survival. And there's some heterogeneity in space and temperature and all kinds of different things that could happen at this biological level, which altogether I think would contribute to what I mean by the emergent properties at this level. So kind of boils down together like all the past and the questions I've been interested in the past few years during my research career. I think it's like how does these kind of context, the ecology evolutionary context, drive the emergent property that we see at the community level? And I'll give you two examples here. The first one is through the ecology of microbial community assembly, and the second one will be about the evolution of symbiosis. All right. So if you Google biodiversity or aquatic community, this is probably the first picture you will see. I think everyone will appreciate that this is a highly diverse pictures of organisms where you have different morphologies, and it's a aquatic arthropod. I'm sure people in the audience will know much better than me. I don't recognize these species, but I can appreciate diversity here. And I think one question that's been kind of proposed is that how come that they can all coexist, and how come they kind of get together and then they can kind of happen to be at the same place? And the idea is that given the environments, there's not many resources or not many types of resources can support this diversity. How come they can still coexist like this? And of course, I'm not the first one thinking about this, right? Like George Hutchinson in the 60s, he kind of framed this as famously as the paradox of a plankton. The idea is that how come is it possible, right? Like these great number of species can all coexist together, despite that these are not very homogenized environment, not very heterogeneous environments, and also there are not many resources that can support them. And one way we can kind of frame this question is using the ideas in kind of systems biology. So let me walk you through these examples. So imagine, we can use this kind of reductionist approach to understand how a property of a system is emergent or not. So imagine as I am a biologist, right? Like I'm sure most of us are biologists. I have this radio, And radio is a box, which is a predates modern mobile phone and other fancy technologies. It's a box supposed to play music and play radio channels. It's kind of like materialize a podcast. Um, and I have this radio, and I know the function is supposed to play some sound or music, but someday it's broke. And I don't know how to fix it. So as a biologist, one thing I would do first, without any energy engineer training, is I would go to find secure funding through writing a proposal, and then I will buy a lot of identical radios that they can be equally functional. And then I'll start to kind of crack them open. And then what I see on the right is there are a bunch of different objects that I can describe then, the shapes and the colors. And I will start by removing them one by one. And eventually, over years of hard works, or decades of hard works, I will have a catalog of how the consequences of removing one or two objects and how that affects the function of the radio. And the question here is that can all this information we work so hard to collect help us repair radio? I think sometimes I think it could if the function is so simple, like the power cord is cut, then replace that part will fix the radio. But I think it will be really tricky if, two or three or more objects have to be tuned all together in order for it to work. And if that is the case, then I would say, that is not trivial, and then we need to account for the interactions among these objects, and therefore it's an emergent property to some extent. So we can use this idea to frame the coexistence question that I proposed a little bit earlier. We can frame it as whether it's an emergent property or it's a pairwise phenomenon. And here's some definitions. So for the system we have here, so the system is of course a coexisting community that we know the properties of coexistence. And the simplest units that can have coexistence is a pair, right? So you could either have a pair that coexists or exclusion, which is like one species beats the other one. And one idea based on the pairwise phenomena is that if all the pairs of species can coexist, the three of them altogether should coexist. Very straightforward. So it's not emergent. On the other hand, it could be an emergent property, right? Like, for instance, the rock, paper, scissors. None of the pairs will coexist. The rock will beat the paper. Sorry, the rock will beat the scissors. But altogether, they will form these loop that can all coexist. So in this case, we will say coexistence at the three species level is an emergent property because none of the pairs can coexist. So to kind of test this idea, in my PhD lab, before I joined the lab, they assemble these communities that provide a perfect playground to test this idea. So the experiment is pretty straightforward. So let's say you have an experiment, you assemble communities in the lab coming from a very diverse environmental samples, and then you introduce them to the sugar water, right? Like you have hundreds of these communities, and you let them self-assemble. And over 12 dilution cycles, if you've ever done any microbiology experiments, it's very straightforward. You grow them, you put these soil substrates in the sugar water, leave them in the incubator for 2 days, take small liquids, inoculate that to the fresh media, wait for 2 days, and repeat that again, and for 12 cycles. Eventually, you will have a community that kind of stabilized. By that, I mean they will always reach a similar community composition at the end of each dilution cycle. So once we have these stable communities, hundreds of them, and we can start by isolating individual communities. So we have the constitutional pairs. And from these constitutional isolates, and then we can reconstitute the pairs and then grow them in exactly the same environments as the community. So we're comparing apples to apples. We control the abiotic environments. And from there, we did same dilution cycles and characterize their outcome. It's either coexistence or exclusion. Just a little bit techy here. this is before Gen AI or ChatGPT, and then in principle, we can, since these are isolates based on morphology, so we could distinguish their, we can measure their abundance by just counting the colonies. So old techniques, but new use. So we could identify the relative abundance of each pairwise competition, and then determine whether the pair coexist or not. All right. So these are the main results. So what you're seeing here on the x-axis is we have the 12 of these community replicates, and from smallest to the largest. And what you'll see here is by the color, if the result is exclusion, you'll be red-ish. If it's the result of pair is coexistence, it's blue, or there's some pairs that we cannot conclude the results. So if the hypothesis of pairwise phenomena is true, then we expect that we'll see a lot of blue here. And that's not what we see. We see a lot of red. So which means that coexistence in these communities is already emergent property, right? Like because the pairs are mostly exclusion. So which led us to the second hypothesis. If it's an emergent property, we expect that they form these rock, paper, scissors loops. So again, we align the x-axis from the smallest to largest communities, and then kind of like a tennis tournament, we can rank each isolates based on how many wins or losses it has, and then from the top one to the lower rank one. That's the y-axis here. And to our surprise, like we only see hierarchy, right? Like the good players or good isolates always beats the lower performing isolates in the same community. So which really rejects our hypothesis of rock, paper, scissors. All right, so what should we do? So in this case, we reject the first one, it's a pairwise phenomena. We also reject the second one, the rock, paper, scissors. And what we see is instead is a competitive hierarchy, right? The good player or good isolate always beats the lower rank one. For the sake of time, I don't have time to talk about this, but we do know some mechanisms that would suggest why this is the case. Feel free to ask me after the talk. But from here, we can already conclude that community coexistence it's an emergent property. And I think this is a good news for ecologists, right? Like it's kind of answer one of the questions been kind of lingering in the fields of ecology for decades, but it's a bad news for people who aspire to engineer microbes toward the function they need. And I think that's one way, like we are not losing hope in the sense that we can still kind of engineer these community functions, even though these functions are emergent property. And one of the answers lies in how we can engineer them, right? Like these ideas are kind of inspired by artificial selection, which, as many of you have known, like the great diversity of dog strains or different livestocks or many crops are all the results of artificial selection by our ancestors. And these are the works that the person who did this, they did not know anything about genetics, they did not know anything about genes, and they did not know anything about how different components of genetic genomics or the genome interacts. They can still do this. And kind of using this idea, many people would kind of think of, okay, if this is the case for microorganisms, then it should work for microbes or microbial communities. Kind of fast forward here. So that's the thing since the first work from 2000 by Swenson et al. On uprights, they aim to kind of engineer microbes that can improve the plant's biomass. And since then has been like dozens of work that kind of try, or it's either microbes or host-related microbes, try to engineer microbial communities to the function they want, like all these variety of different functions, including myself. I did that. I did a trial as well, and the best can be only say it's a modest results. Like it works, but it starts to work at some points. And one reason I think it's kind of like get us scratch our head, and then we didn't know why this is the case, why didn't it work? The principle is so straightforward. And one kind of answer we found is from this old literature by Lewontin in the 70s. Basically, what he was arguing is the any levels of biology can beat under selection. And in here, he referred to natural selection. So the idea is any of these biology can be under selection as long as it fulfills three rules. Like the first one is that it needs to have some variation in the phenotypes. It could be morphology, physiology, or behavior, etc. And these variation should be tied to fitness, right? And this fitness is heritable. And luckily, because we're doing artificial selection, so we as a breeder or the person who are engineering the microbiome can determine which phenotype has the highest fitness. So we only got two rules left. So how do we kind of execute this? And one thing that I think makes the main difference between microbes and other organisms like animals and plants is that microbes here, I mean bacteria, they are sexually reproducing organisms, right? So for the sexual reproduction, they by nature recombination between gametes will generate new genetic variation, whereas asexual reproduction is basically the clone of the same genome. So it kind of loses one main mechanism that would generate variation by nature. So that's one big problem we have if we want to engineer microbiome. The second one is selection has to work on variation. And often the case, if you go back and look all these literatures who try to engineer microbiome, this is the same problem we all face, right? Like selection works in the first few rounds when the variation is still abundant, but it stopped to works at some point because there's not much variation for us to work on. And at this point, then we have no way to select that because there's no variation. And one thing, luckily, that we can do is that these are microbial communities, right? Like we can introduce variation, not by sexual reproduction, but by perturbation. Like it's an ecosystem. And by that, we know, you can perturb the ecosystem by spiking a species, or you could remove some species in order to change the composition of the community, which in turn would change the function. So that's why we kind of find would be a possible solution to this problem of low variation in the selection of microbiome. All right, so back at the time, it's 2020, and let's COVID start to happen, and we're not allowed to do lab work. So that's the time when most of us start to do some more computational work, and luckily that's the good choice because otherwise I won't be able to finish my PhD on time because this is a lot of work if I have to do it in empirical studies. So we kind of designed this Python made a package that in principle it can simulate the protocols of community selection based on simple consumer resource models. I'll show the math here just to give you an impression that this is based on very simple assumptions, and happy to talk more about the underlying equations that make this work. But idea is that we can simulate the protocol you have seen so far, like select then, and then they have variation, and then you work on the selection. You select those communities that have functions you want, and then repeat that cycle again and again. So we have this tool, and here's one example of what it is like to simulate these community selection protocol. So here on the right, the x-axis is selection rounds. It's in silico, so you can do as many as you want. So find out 20 or 40, which might be equivalent to months or years of work in real life. And the y-axis is any function that you can define. For instance, you could be the biomass of the total community, or could be some metabolites that this community produces. And each of these line here is a replicate community. We start with 100. And what I highlight here in solid black line is one community that's at the end of 20 selection rounds, it becomes the best one. But if you go back and trace the first few rounds, it was a mediocre. Um, and if you see the best community in the beginning of the selection, it kind of becomes mediocre later on. And the reason is that, again, this is the community, right? They are dynamic system. They don't settle right away. They need time to stabilize into a stable state. So one thing that we realize is in the previous protocol, most of the protocols kind of jump the gun. We didn't think about like, it need to stabilize before we make selection. So we, in silico, we did this. We allow them to kind of stabilize first. And at the end of these stabilizations, we do one of the perturbations, like here we knock in a species, so kind of perturb it. And here we don't need to use all the communities at the end of these selection stabilization. We only need one, like that is the best one because it's stabilized and we can replicate them. Right? Like in experiments, it's kind of like you take one communities and then you replicate that into 100 new fresh media. And for each of these replicates offspring, you perturb them differently, right? With the hope that the best performing community is already closer to a higher performing peak in the landscape, in the fitness landscape. And in that case, we would be able to find a better performing community after perturbation. And that's what we see. When we perturb the replicates of these top performing communities, some of the offspring become better and some of them become worse than the parents. But that's fine because we only need one that perform the best because we allow them to stabilize. So we don't need all of the communities under selection to be good. We only need one that we can keep on performing perturbation and selection. There are more in this paper that we try different perturbation protocols and different regimes of selection, and they kind of all boils down to these simple rules of we stabilize then, perturb, and stabilize again. So kind of using this idea inspired by artificial selection and directed evolution in protein engineering that we could harness the function of microbial communities. So kind of like to summarize this part of the talk, we could say, hope I convince you that coexistence or microbial community functions, at least coexistence is an emergent property. And even though that community has an emergent property, we can still leverage some principles from evolutionary biology to help us engineer or design the function of the community that we want. All right, so that's the first part. I'm gonna kind of, for the sake of time, I don't want this to be a long talk, so I will skip some slides for the next few parts. But the idea is that I want to show you the other examples of emergent property besides microbial community assembly. All right. So at the point when I worked on this system in my PhD, I got more interested in host-related system, which I will argue also has some emergent property. So when I was drawing these cartoons for like symbiosis with a host and many symbionts, I cannot help to have this image in my head, like the molecule of water. It has one oxygen and two hydrogen, and they're connected by covalent bonds. So to me, I think that it's very similar in essence that as a whole, the system has a function that individual components cannot have. So in this case, I will argue that symbiosis, you can view it as a system with a host and symbionts and many different microbes all together with their interactions will give rise to a function that neither one of the components can achieve alone. So from here, kind of shift gear to like think about symbiosis and to think more about the traits of the symbiosis that manifest by linear interactions between these organisms. So this is very different from the first part of the talk, but that's the question I've been kind of interested in recently to ask about the traits diversity. And I think one question in these world is that we could think of the traits as outcome of different evolutionary paths, right? Whether the traits differences we see from between individuals can be explained by between species differences, right? Like different tree species, different fish species that differ in their morphology, different traits. But some of these, as we can see from the picture of the dog here, like they're all by definition the same biological species, but they differ in their traits a lot. So when we think about like the traits that is so diverse in nature, they could come from these different levels of biology, right? Like you come from the macroevolutionary level or coming from the microevolutionary level. So one kind of fundamental questions in I think in quantitative genetics is to ask to what extent that these traits variation that underlies all evolution processes, is it coming from the level above or under species level? In other words, is it between species or within species? So kind of to tackle this question, started to work on these amazing plant species. It's a legume, Medicago. And this legume, it has very diverse species across the world. For those who don't know, Medicago genus has many kind of like model systems in different fields, right? Like Medicago sativa is alfalfa that you probably have eaten, a couple times in your life in the salad bowl. Medicago Chang-Yu is a model system in the molecular mechanisms, in studying these molecular mechanisms between plants and microbes. And there are also other like ecologically very relevant species like Lupulina, Polymorpha. They are not globally distributed. And from here, we kind of curate a source of 9 species in Medicago, and each of them we have 3 genotypes. So we do have above and below species level variation that we can work on. And what's nice about this legume is that, as many of you have known, legume forms symbiosis with rhizobia, where they exchange nutrients. So it's a nutrient exchange mutualism. Rhizobia provides fixed nitrogen, and the plant provides photosynthesis or carbons to rhizobia. And the organs that are formed, is called nodules in the roots, can be visible by naked eye, and then we can quantify that traits as well. On the other hand, hosts are not just exposed to mutualists, but also some parasites. So in this case, the protagonist is the root knot nematodes. It's a parasite that forms galls in the roots, so it's also visible by eye. You can tell them apart by morphology. And one kind of anecdote about this system is these nematodes, it's a very generalist, so it can infect many, many different plant species. And one thing about their infection mechanism is, I think it's hypothesized to be coming from kind of horizontally transferred from other mutualist microbes, maybe from rhizobia to nematodes, so that it can infect the plants and suppress the immunity. All right, so with this system, we can do some kind of classic manipulations across these panels of 9 species and genotypes, 27 genotypes in total. Classic factorial design in the lab, we grow the plants. They're pretty tiny, so we can grow them in a controlled environments where we can manipulate the symbionts. Either we inoculate symbionts or not, right? Like with nematodes or with rhizobia or not. A total sum of 1,000 plants. Right. So here's the tricky part. So experiment is pretty straightforward. And the way we can kind of decompose the total trait variation we see toward different components of either species or genotype level is the use of a kind of extended linear mix models. So I'm gonna break it down for you, and then that's the kind of core analysis of these work. So suppose we have a trait value, and then it can be decomposed to, first of all, the microbial treatments. It's either rhizobia or nematodes with and without. So that's very straightforward to model. But the variation could also come from, for instance, the differences between species, the host species, and also the differences between host species in response to the symbionts, right? Like by nature, the plants itself can grow without the symbionts, but to some extent they will vary in their morphology. But also these response to symbionts also matter, and then it can create these induction effects. And not just species level, there's also genotypic level where which can also respond in the same way. And then the variation among these genotypes can also contribute to the variation in the traits. And also we include the phylogeny across these species. It's essentially covariance matrix among the species. And of course, the blocking factor, that's when we design the greenhouse experiments, and everything else that we cannot capture by these terms are residuals. So you might be familiar with this. It's a very classic linear mixed model. And then the nice thing about these framework is you can start by decompose the total trait variation toward each individual components and ask which one contribute more compared to the other. And the traits we measure here, there are two kind of main kinds. One is that on the left, the yields, the biomass, the chlorophyll rates, or the leaf size and shape, etc. And these are the traits that the host will grow nevertheless without a symbiont. Whereas the nodules or galls, these are organs that are de novo when the host meets the symbionts. So we can kind of classify the traits in this way based on their genetic composition. And yeah, so here's a picture of the organs. You can see the galls and nodules. They're kind of different. If they look the same to me, but trust me, if after staring at these for I don't know, hundreds of hours, you start to see the differences. All right. So here's the main figure of a result. So one hypothesis that we have here is for those traits that are mostly determined by the host genetics, we will expect that's where the host species differences will set the difference. Whereas for those de novo traits like nodules and galls, because they are only expressed in symbiosis, and then they have a large components influences by the symbionts. So we expect that relatively it's more influenced by the genotypic differences across the host. So if these traits variation is mostly explained by species level, you'll see the red blocks. If it's mostly explained by intraspecific variation or genotypic variation, you'll be blue. So here's what we see, at least for these 4 traits. I'm still working on the other traits, but for these canonical traits that we can identify, we can tell that's biomass or SPAT, it's a chlorophyll reads, chlorophyll content in the leaf, is mostly dominated by species differences. There are some inferences by the rhizobia or by the nematodes, but it's minor compared to the already species differences between these hosts. Whereas nodules and galls, these de novo traits are most relatively dominated by still, species differences, but there's small genotypic variation contributing to these traits. All right. So kind of like sum of this, right? Like when we think about the traits and how especially in this case, the symbiotic traits, which is the outcome of symbiosis between organisms, and how we decompose them, and then we have to carefully account for the history of the microbes and the hosts. So in this case, some host traits like biomass or these host intrinsic traits, we found that it's mostly explained by between species differences. Whereas those traits that are within influenced by both the host and the symbionts, we found that within host species differences can be a more important contributor to the variation. All right. If you kind of fade out for all of the what I talk about so far, this is the only slides you need to remember. Uh, I think the argument I'm trying to make is the species are not independent, and by that I mean they have ecology with the other critters, and also the background environments that will influence them, and also they are bounded by their evolutionary past. And because of this, they give rises to the emergent property at a community level. And I hope I have convinced you, or at least provides two examples to kind of support these arguments, one by the community assembly, the other one by symbiosis. Um, so when I think about symbiosis, I think this is one of the most interesting pop science book that I've read in the past by Ed Young. It's called I Counting Multitudes and the Microbes Within Us and a Greater View of Life. It's already 10 years ago. I read it when I started my PhD. And I like the title, but I didn't know what it means until I was talking about this one day. And then a audience in uh, one person in the audience kind of told me that this is one of the poem that we read in the U.S. high school by Walt Whitman, and it's called "Song of Myself," and it's wonderful. It's a very long poem, and one of the poem reads like this: "Do I contradict myself? Very well, then I contradict myself. And large, I contain multitudes." I think this is a very nice and profound term. And when I think about these, I feel like these kind of resonate pretty well with the idea that when I studied biology, which also contains a lot of multitudes, right? Like ecology evolution and multiple levels of biological organisms. And similarly, the people or scientists who study biology also contain multitudes. And we also come from different trajectories and backgrounds, and which will give rise to diversity of the studies that's been so interesting and captivating in the field. So kind of with that, I want to thank people I've been fortunate to work with in the past from the Wood Lab in Penn and the Sanchez Lab at Yale, which I already moved to Salamanca in Spain. Um, with that, I'm not done with my talk. So I want to kind of take 5 minutes to talk about what I want to do in my current lab since I just started for about now it's like 9 months. Um, so one kind of central question that I have is kind of started from this idea of when I think about like host symbionts relationship, the traditional way, or to think about it, like the first image you have, or people in the field will study them by inoculating one microbe to one host and ask, the outcome of that and try different ways. But in reality, we all know that one host can interact with so many different microbes, right? Like, so the reality is that all these microbes in the same host, they are so diverse. And how do we account for that? So kind of like taking together the idea of ecology and evolution in this context of symbiosis, I think in this case, there are multiple levels, right? Like the plant host within that, so there are bacteria hosts, bacteria symbionts, and within that, many symbiosis streams are in mobile genetic elements. So creates these unnatural levels of biology. So some people in the field call this symbiosis within symbiosis, and they have these, very straightforward and metaphor using the Russian dolls. But I think this is not so accurate in capturing the reality. So when we think about these different levels of biology, and when each of them has interactions with the other individuals, organisms, populations, species, they create these kind of messed up image of kind of like a messed up Russian dolls, right? Like each levels will interact within the levels, but also interact between levels. So that's kind of like the whole, I think, a more holistic image of what actually is going on, even though it's still very simplistic um, uh, kind of image of what the biology, the real biology has. So the kind of study you have these mindsets. I was very lucky. This is a plant I showed David when he visited a couple of months ago. This is a native legume plant called Grona. It's a genus. There are some native species in Taiwan, and two of them are small plants um, that you can find in the lowlands, like almost everywhere. And especially now it's in the summer, they're kind of everywhere. And they're tiny. That's the scale bar of my water bottle here. And then they co-occur, right? So that's an interesting question for like they are related species and they grow basically the same sites, and they are very easy to manipulate in the lab. Here's an image of the greenhouse plants that my students and assistants been working in the past few months to grow them. I mean, they're now kind of out of control, they grow too fast. So kind of using this system, we can ask some very interesting question. Just gonna give you overview, and feel free to ask me afterwards. But some of the questions we can ask in this system is how what drives the assembly of this symbiont community. It could be contingent or deterministic, right? Like by this we can employ the natural variation in the field and then ask how repeatable is this assembly driven by the host specificity. Or we can do that in the lab, right? Like the replaying the table of life style experiments and ask for the same replicate plants, how likely that all these community assemble to the same states. And the other kind of avenue that I've been very excited about and is taking advantage of the materials we have here is by asking whether these symbiosis, how does it adapt to the rapid urbanization? And we do have these kind of nice gradients of urban environments in Taipei Basin, from urban to rural samples, which is often the case when people study urbanization. You have these gradients in terms of space. But what it lacks is usually the resolution in time, right? And we are fortunate to have these Siberian samples from 100 years ago during the Japanese time. And that's a good kind of, and also like over the time, the specimens that we can use to kind of compare whether the modern population, whether it adapts to the novel environments, like the urbanization happens rapidly in the past few decades, and how does that influence the plant's adaptation? Is it based on standing variation, or there's de novo mutation that happens helps the evolution novelty? And last one, which is a little bit more conceptual now, we can also think about symbiosis as it's a multi-partner symbiosis, right? Like, and then when we start to think about specificity, it becomes weird, right? Now you can no longer count how many symbionts that one host can form symbiosis with, because essentially all hosts has a multiple symbionts, and they do have interactions. And in that case, how do you define specificity in this case? And one of the ideas is based on the idea in evolutionary biology by quantifying the epistasis at the level of ecology and genetics to quantify specificity in this case. My last slide is a very long talk. We're still recruiting, and it's a new lab, so we do have fancy space. And we are in a nice greenhouse building surrounded by nature. And you can see from my screen, like the background, that I have the window that's facing the forest. So I really like the space here. So yeah, with that, I'm happy to take any questions.